The shroud is an image of a crucified man with wounds that correspond to the brutality that Jesus of Nazareth experienced.
So why do you think it's real?
They cannot explain how the image is in the shroud. Do you have your phone with you by chance?
Yes, I do.
I want you to do an exercise on camera with me, if you don't mind.
Okay.
Look at the face of the shroud right here.
Run a Da Vinci painting through a NASA depth scanner, and you get a flat, distorted smear. Run the Shroud of Turin through the same machine, and a real human body rises out of the linen in full three-dimensional depth. No painting on Earth does this. And buried inside the same fibers, scientists just found DNA from 5 continents that no medieval forger could have placed there. What that cloth is recording shouldn't be possible. And the deeper they dig, the worse it gets for every theory we have. The night the face appeared, May 28, 1898, Turin, Italy. A lawyer and amateur photographer named Secondo Pia is granted rare permission to photograph the relic during a public exhibition. Late that night, alone in his darkroom, he lowers a glass plate into the developing chemicals. As the image rises out of the solution, his hands start shaking. He nearly drops the plate. What's forming in front of him isn't supposed to exist. On the photographic negative, where light should turn dark, and dark should turn light, a sharp, high-contrast face appears. Deep-set eyes gently closed, a broken nose, a forked beard, bruising along the right cheek. For the first time in history, the figure hidden inside the linen isn't a faint shadow. It's clearly a man, calm, dignified, marked by extreme suffering. A normal image behaves predictably as a negative. Light flips to dark, dark flips to light, and the result looks distorted. The shroud breaks the rule. Photographed as a negative, it appears as a true photograph of a real human being. What the naked eye sees on the cloth is already a negative. 8 centuries before the camera existed, somebody, or something, produced a flawless photographic negative on linen, with no way to test or confirm the result. No artist alive in the Middle Ages possessed the knowledge to even conceive of such a thing, let alone execute it. That was the first crack in the wall. The shroud didn't behave like a painting, it behaved like a photographic plate capturing a single instant in time. And it would take another century before science figured out how violent that instant actually was. The DNA that shouldn't exist. In 2015, a team of geneticists led by Professor Gianni Barcaccia received unprecedented access to the relic. Their goal wasn't to find the DNA of God. Science has no framework for what that would even resemble. They wanted to rebuild the life story of the cloth itself, where it had traveled, who had handled it, whose hands had carried it through the centuries. Using sterile micro-vacuum devices fitted with ultra-fine filters, they pulled microscopic dust and organic fragments from deep between the warp and weft threads, areas where ancient material can stay locked in for thousands of years. They focused on mitochondrial DNA for a specific reason. It exists in hundreds of copies per cell. It survives far longer in ancient material than nuclear DNA. And it functions as a biological GPS, a dependable marker of where a person's maternal line came from on the planet. For weeks, the computers ran, decoding millions of nucleotide sequences and comparing them against global genomic databases representing populations across the planet. Then, the maps appeared on screen. Haplogroups, ancestral lineages, geographic origins lighting up across the globe. The lab fell silent. Nobody spoke. Barcaccia's team was staring at results that contradicted every prediction they had made. This wasn't the genetic signature of one person. It wasn't even the signature of one population. It was something far more intricate, something nobody had foreseen. The findings, published in the highly respected journal Scientific Reports, exploded across the scientific community within days. The expectation was simple. If the shroud were a medieval forgery from France, European DNA should dominate. If it were authentic from Jerusalem, Middle Eastern DNA should dominate. Neither happened. First hit. The Middle East, haplogroups, linked with the Druze, an ancient historically secluded community in Israel, Jordan, Lebanon, and Syria, whose genetics have barely shifted in 2,000 years. Second hit. Western Europe, haplogroups, U5b and H1 through H3, consistent with the Poor Clare nuns who restored it. the House of Savoy who possessed it, and the pilgrims who pressed close to it for centuries. Expected. Third hit, North and East Africa. Haplogroup L3, Egypt, Ethiopia. Direct contact with some of the earliest Christian communities on the African continent. Fourth hit, South Asia. Haplogroups M39, M56, and R8. Markers typical of the Indian subcontinent. Now the team is staring at each other. Fifth hit, East Asia. Haplogroups D4 and G2A, China. Genetic markers from a population separated from Jerusalem by more than 4,000 miles of desert, ocean, and mountain range. India, Africa, Europe, the Middle East, East Asia, all encoded into a single ancient cloth. 5 continents of human ancestry, woven into the fibers of one burial linen. They reran the controls, they tested for lab contamination, they sequenced again. The signal held every time. If this is already pulling you in, hit subscribe right now, because the DNA was only half the secret. The other half is something nobody saw coming, and it points straight to a crime scene in Jerusalem. A world written on cloth. No medieval craftsman in a French abbey around 1350 could have collected genetic material from China, India, and East Africa. Globalization didn't exist at that scale, and no forger could have planted those traces in a way that would deceive geneticists 600 years later. The DNA on the shroud isn't random contamination. It's a shared biological memory. When Barsacha mapped the markers geographically, they aligned almost perfectly with the ancient Mandylion route, a path scholars had long dismissed as legend. According to early Byzantine and Syrian records, the shroud was folded so only the face showed, and carried as a relic for centuries. It didn't begin in Europe. It began in Jerusalem, then moved to Edessa in the 2nd century, a city that sat directly on the Silk Road. That single fact explains everything. Caravans from China loaded with silk, merchants from India carrying spices, Persian diplomats, Arabian traders, pilgrims from every corner of the known world, all passing through Edessa, all coming to honor the image believed to protect the city from invasion and plague. They stood close. They kissed its casing. They touched the cloth. With every encounter, microscopic traces, skin cells, hair, sweat, the dust clinging to their robes from 1,000 miles of road, settled into the fibers. Century after century, the DNA of the entire ancient world collected on its surface like invisible sediment, preserved by the dry interior climate of the cathedrals where the cloth was kept. From Edessa, it moved to Constantinople in 944 AD, the largest melting pot of the ancient world. In 1204, during the Fourth Crusade, the city was sacked and the shroud vanished. It resurfaced in France around 1353 in the possession of a knight named Geoffroy de Charny, the supposed forger's window. By then, it had already absorbed 1,000 years of human contact across 3 continents. This is where the genetic evidence delivers its hardest blow to the forgery theory. The DNA isn't noise. It's a geographic fingerprint no forger could have produced. But the genetics were only half the hidden code. The other half was written in plants. Pollen, blood, and a crown of thorns. Israeli botanist Avinoam Danin and Swiss forensic scientist Max Frey independently analyzed pollen grains trapped deep inside the linen. The same kind of microscopic plant evidence used to convict criminals when the rest of a crime scene has been wiped clean. Frey collected his samples by pressing sterile adhesive tape against the surface of the cloth, lifting away the trapped material a layer at a time. They identified 58 different plant species, 17 native to Europe, expected. The majority came from elsewhere, the Middle East, Turkey, the Anatolian steppe, perfectly tracking the ancient route through Edessa. Then came the species that grow nowhere else on Earth, except a narrow corridor between Jerusalem and Jericho. One dominated, Gundalia tournefortii, a thorny desert thistle. Its pollen made up nearly half of all samples, concentrated heavily around the head and shoulders. Why would thorn pollen saturate a burial cloth around the head? The crown of thorns. Gundalia tournefortii, with its long needle-like spines, is exactly the plant Roman soldiers could have twisted into a mock crown. It blooms near Jerusalem in early spring, precisely during Passover, A second botanical clue came from Zygophyllum dumosum, a plant native only to the Judean desert and parts of the Sinai Peninsula, found in notable quantities. No medieval forger working in France could have collected pollen from plants endemic to Israel, let alone spread it invisibly at a microscopic level across a piece of cloth. A forger would have used paint. Pollen cannot be painted. The blood told its own brutal account. For decades, skeptics argued the reddish stains were ochre or tempera mixed with gelatin. That claim collapsed in 2017. A team led by Professor Giulio Fanti at the University of Padua analyzed the stains using transmission electron microscopy and Raman spectroscopy. At the nanoscale, they found not pigment but blood. Human blood, type AB, one of the rarest blood groups in the world. This wasn't the blood of a healthy person. Inside it, scientists found nanoparticles of creatinine and ferritin bound to hemoglobin. Such extreme concentrations occur under one set of conditions only: catastrophic, fatal trauma combined with prolonged torture, dehydration, and severe muscle destruction. When muscle tissue is repeatedly destroyed, a process called rhabdomyolysis creatinine floods the bloodstream in massive amounts. The blood preserved on the cloth recorded exactly that, a biochemical signature of agony. The man wrapped in this cloth was beaten to a physical condition already incompatible with life before crucifixion even began. Forensic mapping of the wounds shows over 100 individual blows across the back, shoulders, legs, and chest, consistent with Roman scourging using the flagrum, leather whips embedded with lead weights and bone fragments, designed to tear flesh away with every strike, and the blood stayed red, which should be impossible after 2,000 years. Old blood normally darkens to brown or black within weeks. But the samples carried unusually high levels of bilirubin, released by the liver during extreme stress, and bilirubin preserves the red color of blood indefinitely. A painter can imitate the appearance of a wound. No human hand can recreate the biochemical signature of extreme trauma, organ failure, and hypovolemic shock. The test that got it wrong. In 1988, 3 prestigious laboratories, Oxford, Zurich, and Arizona, carbon-14 dated the fabric to between 1260 and 1390, AD medieval, a forgery, case closed. The world accepted it. Even the Catholic Church stepped back, calling the shroud an icon rather than a genuine relic. 3 decades later, researchers found exactly where the mistake had happened, and the problem wasn't the technology, it was the sample. The 1988 piece was cut from the very edge of the shroud, a corner handled countless times, soaked in sweat, oils, and candle wax over centuries. Then chemist Raymond Rogers of Los Alamos National Laboratory uncovered something devastating. That corner had been repaired during the Middle Ages. The Poor Clare nuns had woven new cotton threads into the damaged edge, dyed to match the aged linen. The main body of the shroud contains no cotton at all. The labs didn't date the shroud, they dated the patch. In 2022, physicist Liberato De Caro at the Institute of Crystallography in Bari ran a completely different test: wide-angle X-ray scattering, or WAXS. Instead of measuring contaminants, it measures the aging of linen cellulose itself at the atomic level. Over time, the polymer chains slowly break apart under exposure to radiation, humidity, and temperature. This process functions as the material's internal clock. And unlike carbon dating, it cannot be skewed by surface contamination, candle smoke, or medieval repairs. De Caro compared the shroud against fabrics of known age, from Egyptian mummy wrappings dating to 3000 BC to medieval European textiles of every era. The shroud's molecular structure didn't match medieval fabrics at all. It was much, much older. It matched linen recovered from the fortress of Masada in Israel, dated between 55 and 74 AD, the 1st century, the exact time of Christ. In 1988, science dismissed the miracle. In 2022, it brought it back. But one mystery still rises above every other, and modern physics cannot explain it: the impossible image. No brushstrokes, no pigments, no ink, nothing. For decades, scientists looked for the medium that produced the image and found nothing. There is no paint to scrape, no dye to dissolve, no binder holding anything to the surface. The image exists only on the outermost 200 nanometers of the fibers, hundreds of times thinner than a human hair, thinner than the wall of a single bacterium. Strip away that microscopic layer, and the image disappears completely. It's a chemical transformation caused by oxidation and dehydration of the linen itself, a scorch left by an unknown source of energy that altered only the topmost molecules of the cloth, leaving everything beneath untouched. Scientists at Italy's National Energy Agency, ENEA, spent 5 years trying to reproduce it under controlled laboratory conditions. They tried acids, they tried heat at every temperature, they tried gamma, radiation. Nothing worked. Only a brief, intense pulse of vacuum ultraviolet radiation came close to matching the chemical signature on the cloth. And even then, only on a few square centimeters. The technology required to produce such a pulse on the scale of the shroud didn't exist anywhere on Earth in the ancient world, and it didn't exist in 1350 either. To imprint an image across nearly 4 square meters of fabric, the body would have had to release a coordinated burst of energy lasting less than a billionth of a second, powerful enough to mark the cloth at the molecular level, precise enough not to burn it, a flash of light shorter than a billionth of a second, etching a perfect human form into linen at a depth thinner than a soap bubble. No technology in 1350, in 1500, in 1800, or even today can replicate it. And then there's what NASA found in 1976. 2 scientists at the Air Force Academy, Eric Jumper and John Jackson, fed a photograph of the shroud into a VP8 image analyzer, a device built to convert light intensity in spacecraft photographs into 3-dimensional terrain maps of planetary surfaces. They expected the same flat, distorted result you get from any photograph of any painting on Earth. Instead, the screen rendered a perfect 3-dimensional human form. The image contains accurate spatial depth information at every single point. The intensity of the imprint corresponds exactly to the distance between the body and the cloth. Closer points darker, farther points lighter, in mathematically precise proportion, not approximately, exactly. Run a Da Vinci painting through the same machine and you get a flat blob. Run the shroud through it, and a real human body rises out of the fabric in full 3-dimensional depth. No painting on Earth does this, no photograph does this. No artist, ancient or modern, has ever managed to recreate it. Digital analysis identified shapes over the eyes consistent with coins, matching rare lepta minted. Under Pontius Pilate, around 29 AD, these coins circulated for a brief window in a single region. The probability that a medieval forger could have known about them, let alone reproduce them at the right size in the right place, is essentially zero. The nails pass through the wrists, not the palms, exactly as forensic science predicts for crucifixion. Every medieval painting gets this wrong, every single one. Artists place nails through the palms because that's what they believed and what every other artist before them had painted. The shroud shows the anatomically correct location, the only place a nail could actually support the weight of a hanging human body without tearing through the flesh. And the thumbs are retracted into the palms, a nerve response caused by the nail damaging the median nerve as it passes through the wrist. A detail no medieval artist could have understood, because the median nerve wasn't named or mapped until the 17th century. Layer by layer, biology, chemistry, physics, geology, numismatics, every independent discipline that has ever touched this cloth converges on the same city and the same narrow window of time. Jerusalem, between 30 and 33 AD. Different scientists, different decades, different instruments, different countries, same answer. A medieval forger would have needed to create a photographic negative centuries before photography. Embed pollen from plants found only between Jerusalem and Jericho. Produce blood with trauma biomarkers undiscovered for 600 years. Encode 3-dimensional depth information no modern lab can duplicate. Place coins on the eyes from a Roman procurator who ruled for 10 years and was forgotten by history. And collect mitochondrial DNA from China, India, Africa, and the Middle East, all from a French abbey with a candle and a paintbrush. The Shroud of Turin is not a painting. It's a forensic record of a real human being, a biological archive, a silent witness lying in a sealed reliquary in a cathedral in northern Italy, still encoding information no scientist on Earth can fully explain. Every test we run on it tells us more about what it is. None of them tell us how it was made. So, The question is this: do you believe science will one day explain what happened inside that tomb? Or are some events beyond the reach of any instrument we will ever build? Drop your answer in the comments below. I read every single one. And if you want to see the next piece of evidence the mainstream still refuses to talk about, the lost gospel that may rewrite everything we thought we knew about the resurrection, that video is dropping next. Hit subscribe so you don't miss it.